Starting a new experiment. This video explains how to start and set up a new experiment in SpectroFlow when samples are in individual fax tubes, which are manually loaded on the instrument. The setup for loading samples from multi-well plates or from the tube rack is slightly different, and we have a separate video that explains how to use the autosampler. After logging into the software, click Acquisition, then click New to create a new experiment. Name your experiment and optionally add a description. Next, you will add your fluorochromes to the experiment. Use the Type to Filter box to easily search through the list. Click the Add button. Once you have added all the colors in your panel, click Next to move on to the Group tab. Select the carrier type that you will be using for sample acquisition from the drop-down menu. For unmixing, you will need to add a reference group. SpectroFlow will automatically create a list of reference controls based on the fluorochromes added in the earlier step. Make sure the control type of reference control, which can be cells or beads, matches your control samples. Note the unstained control should always be cells. Adding antigen labels to your samples is optional. In this experiment, we will use reference controls prepared using lymphocytes and stained either with anti-CD45 antibody conjugated with PE or anti-CD3 antibody conjugated with APC. We will also use a reference control made with beads stained with anti-CD8 antibody conjugated with FIT-C. If all our reference controls contain both negative and positive events, we would leave the settings on this screen unchanged. The SpectroFlow software has options for managing situations when reference controls do not contain negative events. For example, in our experiment, since CD45 is expressed on all lymphocytes, nearly all of our cells will stain positive, and we will not have sufficient negative events. For this reason, we will select a universal negative that will be used by the software as the negative population for this control. Click the Universal Negative dropdown and select the Unstained Cells control. Our CD3 APC reference control does not need a universal negative because there are both CD3 negative and CD3 positive cells among lymphocytes. The compensation beads that we use in this experiment will all stain positive for FIT-C, and we will not have negative events in this control. That's why we need to define an additional universal negative control, which will contain unstained beads and will serve as the negative population for the FIT-C control. After defining the additional universal negative, select the negative bead control from the universal negative dropdown on our FIT-C control. Click Save to add your reference group to the experiment. We will wait to add our experimental sample groups until after running Unmixing, so for now, click Next. In the Markers tab, you may label your sample groups with their tagged antigens. We can return to this tab to label our experimental samples later. Click Next to move to the Acquisition tab. The experiment user setting should be set to the Cytec assay setting by default. Your reference group controls should have the default raw worksheet selected as their worksheet. When the percentage of positive or negative events is very low, you may need to increase the number of events to record. You should have at least 200 events in each of the positive and negative gates of your reference controls. For our cell controls, we set the event limit to 10,000 to start. Stopping volume and stopping time may also be used to set recording limits. We will return to this tab later to adjust the settings for our experimental samples. Click Next. The Keywords tab may be left unchanged. Click Save and Open. Let's briefly go over the experiment layout in SpectroFlow. Your experiment name is in the top left corner, next to which is the X to exit your experiment. On the left side of the screen are the group list, the acquisition control for running and recording samples, and the instrument control, which contains the gain information of all detectors in a saved user settings. Lastly, in the top center, you will see your worksheet tabs. The SpectroFlow software saves your experiment in multiple parts: the FCS files of your samples, the user settings file, and the worksheet files. When a change is made to the worksheet or user setting, an asterisk will appear next to the name. This indicates that a change has occurred that has yet to be saved. Please do not save over any default worksheets so that they can remain standard for all experiments. The Cytec assay setting is a user setting based on the system's most recent successful QC and cannot be overwritten. As you change gains to suit your experimental samples, for example, forward scatter and/or side scatter gains, you may want to save a new user setting that can be used anytime you run this specific experiment, but this is not required. Now, let's run our experimental controls. Activate the sample you would like to run by clicking the green arrow next to the tube icon. To load a tube, open the black cover and place your tube onto the sample injection tube for short sit until it clicks in place. The recommended events to display value is 10,000. Set the flow rate to low, then click Start to acquire your sample. It will take a moment for the sample rate to stabilize. Your event rate must be below 10,000 events per second. If the rate is higher at the lowest flow rate, you will need to dilute your sample. If the rate is lower, you may increase the flow rate. While acquiring a sample, the tube icon will turn yellow. While it is yellow, do not remove your sample from the sit. Adjust the forward scatter and side scatter gains to center your population of interest in the corresponding forward scatter, side scatter plot. Adjust the P1 gate to encapsulate the population. While your sample runs, monitor the spectral signature in the spectral plot on the top right of the worksheet. This plot shows the fluorescence intensity of every detector on the Aurora Analyzer. For the unstained control, the fluorescent expression in this plot represents the autofluorescence present in your cells. You can simply record this unstained control without any further adjustments. When the recording limit is met, the sample will automatically stop running. Since the icon is green, it is safe to remove the tube from the sit. When the tube is removed, the instrument will automatically perform a sit flush to prevent sample contamination. If you would like, you may manually stop running the sample at any time by clicking the Stop button. You may also pause with the Pause button. Since we adjusted the forward and side scatter gains, now is a good time to save our user settings. Load the next sample and click Start. Now that we are running beads, we may need to readjust the forward and side scatter gains to see the population in the plot. After recording all our unstained controls, we will move on to the single color controls. We can easily load our previously saved user settings from the drop-down list. Alternatively, you can save a separate user setting for the beads and alternate between the two settings. For your single color reference controls, use the Aurora Analyzer configuration chart to confirm that the emission of your control matches the expected signature and peak fluorescent channel. For example, PE has peak fluorescence emission in the YG1 channel and lower emission peaks in UV9, V8, and B4. Use this technique to confirm that each reference control has the expected spectral signature. You will also need to be sure that your emission peak is on scale in the spectral plot. If the emission peak is higher than the fluorescence intensity maximum, you will need to lower the gain to bring it on scale. To do this, go to the User Settings, click on the laser with the peak emission detector, and add a negative value to the All Channels % box. Continue to adjust this value until your emission peak is on scale. Note, your sample will need to be running to see a change in fluorescence intensity. Once you have found an optimal value, go to every other laser tab in User Settings and input the same value into their All Channels Sent boxes. If the peak emission is on scale for all fluorochromes, there is no need to adjust any gains. Once the gains are set, record the reference control. Repeat this process for all remaining reference controls. When all reference controls have been recorded, click the Unmix button. A new window will open for unmixing. You may adjust the universal negatives for your reference controls if you notice that any of your controls does not have a negative population. Otherwise, in the bottom right, we will select our unmixing method. Since our unstained cell control is a homogeneous sample, we will select unmixing with AF extraction. Further information on the different options for autofluorescence removal will be covered in a separate training video. Click Next. Now, move the gates. P1 should encapsulate your main population of events. Note, if you hold the Control button while moving the P1 gate, it will move for all other controls of the same type, beads or cells. Move the positive gate over the bright peak in the histogram plot. Click and drag the vertical gates in the spectral plot to the detector that gives the highest signal intensity. For maximum unmixing accuracy, gate the top half of the positive peak and the bottom half of the negative peak. This will maximize the purity and the difference between the mean fluorescence intensities of the positive and negative populations. For reference controls that have a universal negative selected, you will not see a negative gate in the accompanying histogram plot. Once you have set the gates for all the reference controls, you may click the Next button to see the spectral profiles of your controls, the similarity matrix, and the complexity index of your experiment. Finally, click the Live Unmix button, and the software will perform the spectral unmixing calculations. You should not click Create New Unmixed Experiment. That button is for creating a copy of the current experiment, which can be used for data analysis only. Unmixing has been calculated. Set your user settings back to the gains for cells to prepare for running your experimental samples. In the next video, we will go over the setup for experiment sample acquisition and gating.